All right, welcome back, everyone. I hope you enjoyed lunch and the stimulating sessions this morning. We've got four, I think, half an hour-ish length talks in this session, where we're going to be taking a sort of disease-by-disease, phenotype-by-phenotype consideration for where we are on variant to function. And without further ado, I'll introduce our first speaker, Tune Pears, who joins us from the Novo Nordisk Foundation Center for Basic Metabolic Research and is an associate professor at the University of Copenhagen. Tune also trained here in Joel Hirschhorn's lab, so we've been friends and colleagues for quite a few years now. I guess we're both getting a bit gray-haired, huh, Tune? Yeah, let's hear about what you're up to on V2F for obesity. Take it away, Tune.
Great. Thank you very much, Ben, and thank you to you, Melina, and Casper for the invitation. It's a great pleasure and honor to be here today. So last week I went to a meeting where I heard a doctor talking about a patient he had met, and that patient had lost 50 pounds of body weight throughout the past year, and she was really referring to it as an out-of-jail card, like the card you can draw in a Monopoly game. So I think many of us it's hard to fully grasp what it means to live with obesity, but I think it's a heavy burden. I'm saying this in part because I think the astonishing development we've been witnessing in the variant to function field has been paralleled by some really remarkable development in the field of anti-obesity drug development. So we are now at a stage where doctors are at a stage where they can prescribe drugs that are really effective. And as an obesity researcher, it's not something that we have been seeing coming that fast. So I'll get back to this throughout my talk. So I would say, technically speaking, the opportunity is pretty clear. And thanks to Melina for mapping it out that clear. So the GIANT consortium and other major genetics consortia, they provide us with thousands of genetic variants that are associated with obesity risk phenotypes. So we only have to now map the regulatory logic because that will allow us to connect those variants to genes, proteins, cellular states, molecular functions, and ultimately back to the risk phenotype that is rendering a person susceptible to become obese, or maybe rendering a person or maybe making a person not able to lose weight on a certain drug, or maybe giving a person a certain side effect. So we have a theoretical path that is clear. However, in reality, there's a sort of key challenge that we face, and it's basically a very simple one. Which cell population should we study? So we know, based on the work from Melina and others, we know that if you take GWAS variants that are associated with body fat distribution, so simply speaking, whether you have an apple shape or a pear shape, if you take those genetic variants and you look at where those variants are likely to do the change that increases risk towards one or the other shape, then those variants mostly fall onto adipocytes. And that basically tells us, we know that body shape is a predictor of many cardiometabolic comorbidities. So it basically tells us, if you're interested in us understanding the comorbidities downstream of a given weight, then the adipocyte is like the perfect system to study. We also know, if you are interested in body weight, so not in shape but in weight, which you can measure, for instance, by looking at or computing the body mass index, which is easily computed, which is why there's a ton of GWAS data for it. If you take those variants, and they have been found by Joel Hirschhorn, Ruth Loth, and others in the GIANT consortium, if you take them, as I did during my postdoc studies, and you simply look at what are the genes that are expressed in these loci, then the picture looks differently. Then you see a strong signal for the nervous system. And since then, a former graduate student in my lab also went through single cell data throughout Human Cell Atlas, single cell data throughout the the human body and for the mice, so just hundreds of cell types, and the picture is the same. Neurons are coming up. Interesting here is we don't see a clear glial signal, and we potentially can talk about that later, but at least, again, neurons are showing up. So to sum that part up, so I think we are at a pivotal phase in terms of obesity research. It's an early step still compared to where the field is at in adipocytes, but we are at a stage now where we sort of have to identify what are the neuronal cell populations that are really regulating body weight. And the key challenge is to find that these cells that we should study. So, and I wanted to talk you through two of our efforts. One is based on some perturbation studies we do in model systems in mice, and the other one is based on human embryonal stem cell model we have been developing with a collaborator. So so we need to get to this stage and need to figure out what cell types to study, sort of to enter this framework where we then are within a sort of, where we have a path forward in terms of leveraging genetics data to understand molecular functions. Okay, so, but first talking about our studies using mice. So if you look at the response and the weight loss pattern in downstream of a given drug, anti-obesity drug, then it's very simple. It's highly correlated between mice and humans. So a basic question we've been working with in our group is to find out whether we can use mouse models to identify relevant neuronal cell populations. And specifically, we're interested in finding neuronal populations that are potentially explaining or mediating the, or important for the effect of these incretin mimetics. So these incretin mimetics are typically GLP-1 receptor analogs that are acting somewhere in the body, and it's still sort of unclear through which mechanisms they are sort of driving weight loss. So what we know, though, is work from the University of Michigan, where Randy Seeley and his group, they had been taking animals, treated them with liraglutide, which is one of these GLP-1 receptor agonists, and then they have obviously, or not obviously, but they've seen then these animals lose weight. However, they had then knocked out the GLP-1 receptor specifically in glutamatergic neurons. And if you do that, or when they did that, then there was basically no weight loss downstream of liraglutide, this GLP-1 receptor analog. So this basically tells us most likely the weight loss lowering effect of these incretin mimetics are mediated through glutamatergic neurons. A former PhD student, Metabolic, was sort of curious to figure out whether we could identify more specifically what those glutamatergic neurons are. And in collaboration with researchers from Novo Nordisk, we dosed animals with semaglutide and then took out 7 brain areas that we from previous work knew would be activated upon sema treatment. We performed RNA, bulk RNA sequencing, and we simply looked at what are the brain areas among those 6 that changed the most. And we identified changes here in the area postremor as sort of the most responsive brain area to the drug. So we simply went ahead and we looked more closely into the hindbrain where the area postremor region is located. And it's a special region because it's a circumventricular region, meaning that the blood vessels are fenestrated, so all the neurons, or many of the neurons in that area, they can sort of extend their processes out through the blood-brain barrier and send circulating signals. So it's one of the interfaces between the periphery and basically the brain, similar to the hypothalamus. We did single-cell sequencing because we, at that point, didn't know what the cell populations in the dorsal vagal complex, these 3 brain areas is. identified all cells sort of that typically are identified in a brain regions, about 10 non-neuronal cells and a number of neurons. Then when looking further deeply, more closely into the neurons, we expectedly identified that there were several neurons that were expressing the GLP-1 receptor. That was known. I mean, their molecular identity wasn't known. People had been doing studies showing that the GLP-1 receptor is there, and now we sort of had the transcriptional profile of of these neurons, and there were 2 GLP-1 receptor positive populations, maybe 3. And also we identified a population that expressed CALS-R, so the calcitonin receptor and RAM3. And that's another interesting receptor complex because it senses changes in amylin. Amylin is secreted after a meal, and it sort of terminates your eating response. And there are, companies have drugs in the pipeline in their phase 2 trials for amylin receptor agonists. So we are at a stage potentially where amylin also becomes an interesting or a viable weight loss drug. So we identified those various populations, and what we then did was to take the genetic association data that is available for body mass index, for obesity, and simply asked, if you take all of those cells and you test whether genes in obesity-associated loci enrich for genes that are specifically expressed in that cells, what do you find? So what cell populations are preferentially expressing genes that are also located in obesity loci? Then we found a very surprising, to me, result because it was very distinct. So here we found we identified 4 populations that are expressing genes that are co-localizing with obesity signal. 2 of them are the GLP-1 receptor populations, so that's maybe not too surprising. It's still very surprising to me that the signal was that clear. It really it jumped out on us. And also it was very interesting that the amylin sensing population, this calcitonin receptor cell type, was the most strongly enriched one. Right. There was a 4th population, IR1, and we looked closely at it. It hadn't been reported before. We went through all the genes that are expressed in that population. We sort of didn't find any receptor that was could register changes in any hormone. So at that point, we sort of didn't really know what that cell type is doing. But it was one of our 4 findings. There previously had been DREAD models, so models where you artificially activate a neuron, focused on the GLP-1 receptor population. And they had shown if you activate those populations, you reduce body weight. So we knew those 2 populations are weight regulatory, apart from them expressing the GLP-1 receptor, which is the target of these incretin mimetics. So we, with our collaborator, Martin Myers from the university, went ahead and he made a rat model because the area postrema is very small. It's hard to have a DREAD where you only activate that population in the area postrema. You need to sort of do it in the rat model where the brain is larger. He made a model that expressed calcitonin receptor in the, so that we could use a DREAD system to activate these cells. And when Martin and his team activated them, we saw that it reduces body weight. So this is sort of, at least in animals, it tells us if you activate those neurons, you reduce body weight, which is sort of in line with the pharmacological data being available that amylin is a strong putative, or it's a good target for potentially treating or inducing weight loss. Now, since we published that work, a student in Martin's lab had been looking more closely into this IR1 population, this one over here, and he had been developing a DREAD model so that we could artificially activate and also inhibit these neurons. This is work in progress, but not too far, a few months ago, he found that if when he activates those neurons, then the animals reduce food intake. And it's not only short-term food intake, but also long-term food intake. Importantly, also, when he activates them, you can do like conditional taste aversion experiments. We see that these neurons are not aversive. And that's very important because we know that GLP-1 receptor neurons in the area postrema, they are in part aversive. When you start taking incretin mimetics, there's a run-in phase because it's not easy just to take them at full dose. You sort of need to get used to it. The interesting and important thing here is these cells seem to be non-aversive. Great, so that's basically, to sum up that part. So by studying, doing perturbation studies or experiments in animals, we seem to be able to identify cell populations that are relevant to obesity. And this we do by sort of integrating the transcriptional responses we see in these models with human or with genome-wide association study data. So currently, we have about 14 different perturbations across the hindbrain and the hypothalamus, where we have a total of over 1.3 million cells, where we are integrating those cells into an in silico model where we see what happens if you dial down the expression of a gene in terms of neuron activity and expression in that cell population and in neighboring cell populations within a given dietary regime, so within a high-fat diet or leptin-treated animals and so on, because we think so we can use those data to get more closely to also what are the pathways for which some of these drugs act. So I briefly wanted to talk about the human studies, the human models we're sort of working towards. So this is the mouse work, which we do in parallel with the human work. I mean, the human cell line work, both in a way as sort of imperfect models of metabolism. In the mouse, we have a physiological system, but it's not fully conserved with humans, but we can do perturbations. In the cell lines, we are having human cells, but obviously they aren't sort of embedded into a physiological system. Anyways, both of them are important, and the work that I'll be showing here was to a large extent driven by my close collaborator, Agnete Kirkeby, who's heading a lab in Copenhagen, and it was done by Zara by Nørgaard, who is a PhD student who just defended her thesis a couple of days ago. So we had we are interested in, and many other people interested in the hypothalamus. So the hypothalamus is a part of the forebrain. So now we are going from the hindbrain to the forebrain. People also refer to it as the sort of heart of the brain because it's very important for multiple different behaviors, including feeding. The hypothalamus is also a circumventricular organ, so you also have this fenestrated blood-brain barrier, so the projections from the cells can sense, again, what's circulating in the blood. And two of the sort of key cell types in this brain area are these AGRP and POMC neurons. For POMC neurons, there have been, there are models that allow you to differentiate iPSCs into POMC-like cells. However, we don't have that model for AGRP cells. And these two cells sort of are potentially equally important, plus additional other acute nucleus or mediobasal hypothalamic cell types that really are important. So Sarah's basically PhD work was to set up a protocol that we can use to generate these AGRP neurons. So after years of work, she was able to produce POMC neurons. So the red stain here is a MAP2 stain, so it's basically a neuron stain, and here she's staining for the POMC peptide. Excitingly, also, after a lot of optimization steps, she's now able to see AGRP neurons. Again, the red stain is the general neuron stain, and the green stain are these AGRP neurons. So that basically tells us we now have a protocol that we think we can use to sort of study and do some of the variant function work that we have been hearing about this morning. And we know that we think these cells are functional. We don't know it fully yet, but when we treat them with leptin, we see an induction in neuronal activity, which makes sense because most of the cells we have in this dish, they respond to leptin by being activated. Not all cells do, but most of them we have in the dish, they do. This is how it looks at the single cell level. So we have a few HERP cells here and a lot of POMC cells. And on top of that, we have a number of other cells that we are almost equally excited about. Several of them express the GLP-1 receptor and also the CALS-R receptor and some other genes that we know are of relevance in metabolic research. In work that Kasper's and my group are doing in collaboration, we have been looking at if you sort of take more generic glutamatergic neurons and you use, I think Noah and Greta use a single-cell DRS like a tool developed by Elke Price, where you basically score for each cell its enrichment in GWAS signal. If you do it, and you sort of plot it on a scale from dark blue to yellow, then we find that there's some enrichment in these more generic glutamatergic NGN2 derived neurons. However, when we look at the hypothalamic cells, we start seeing much more signal. So these sort of early, and here's the comparison just visualized in a histogram. So these sort of preliminary studies or efforts, they just tell us sort of give us an indication that these hypothalamic cells are really capturing some of the genetic susceptibility we think is mediated through the hypothalamus. I wanted to sort of conclude that part in my talk by just saying sort of we are at a stage now where we are able to produce POMC neurons and HERP neurons, maybe the two most important cell types if you sort of were to take a guess here and now in terms of obesity, well, along with the hindbrain neurons I was showing you. We're able to generate those cells now, and it really gives us a basis where we can move ahead to sort of also have this protocol is based on human embryonal stem cell derived cells. We want them as iPSC cells. Greta is working on it in Kasper's lab. We want to understand the regulatory logic because that will allow us to perturb them and profile phenotypes, ideally in a lipocyte profiler way where we just agnostically profile them after, for instance, perturbing the genes that we know from exome sequencing studies are associated with obesity. Because that now allows us sort of to move deeper into this trajectory where you go from a genetic variant to a ultimately risk phenotype that hopefully will be of translational value in humans. So with this, I wanted to conclude my talk and just acknowledge the various people from our lab who did the work that I've been showing. And I also wanted to acknowledge Agnete Kirkeby and Sarah for all the human stem cell derived work and the funding agencies. Thank you.
Questions for Tuna? I might take speaker's prerogative and ask, what about reward pathway biology as it relates to the GLP-1 agonists, given the anecdotal evidence of, potential downstream consequences on things like gambling addiction or narcotic addiction? Do we think those are all wired into the same satiety response or somewhere else? And how do you think about that in the context of obesity?
Right. Yeah, that's a good question. Currently, semaglutide is treating every single condition we have in a human.
So it seems.
So it seems. I think little is known about how that molecule potentially also impacts reward. It seems to do it, whether it's mediated through the hypothalamus or the hindbrain or through the lateral septum or some of the other ventricular areas, we don't know. So personally, we haven't looked into that. I'm quite curious to see how that turns out, but I don't know the answer. Yeah.
No, I just want to say, wonderful talk, Tone. and make a statement of how transformative I think this is in terms of having a hypothalamic neuron system that appears to enrich in signal for genetics, which in the community currently, there is no such model at all, right? So I think this is a leapfrog towards being able to trace down variant effects from a large fraction of the obesity-related risk variation. I'm super excited about this. Beside leptin, did you try any other, and that goes a bit, on Ben's question, did you try any other stimulatory conditions on these models yet and test outputs? And what other type of phenotypic characteristics are you trying to evaluate to benchmark further and to iterate on differentiation protocols and so forth to improve further?
Right. Thank you. We are looking at neuronal activity, and they respond to potassium chloride as they should. We haven't looked into semaglutide, cagrilintide, this amylin receptor analog, ghrelin, insulin, glucose. There's a number of sort of stimulants that we know are modulating activity of these neurons. So it's very fresh data. We haven't been doing all of these experiments. Hopefully they will respond as they should, but we'll see.
All right, any other questions? All right, let's thank Tune again for a lovely talk.